I'll kick off by, I think, talking a little bit about, analysis of extracellular RNA is a very active research area, as we all know. I'll give in this talk a very quick intro, given that I'm the first up, into what it is that we're talking about when we say EVs and exosomes, and why they're useful, especially for molecular diagnostics, and then talk about open problems. And there are more than this. This is just going to be a brief overview, but in my mind they fall into two major categories. One is that the sample and RNA quality and the way that you isolate extracellular vesicles is quite variable, and introduces variability of its own. And I'll talk a little bit about issues that we have with identifying likely origin of these vesicles in the RNA. If you're hoping to find a biomarker for a disease of a rare cell type in the human body, then it's challenging because you have contributions from all over the body into various different biofluids. So I'll go through that in a minute. But so first up, there's been a pretty big shift over the last decade or so in terms of molecular diagnostics. The first is that we're moving away from morphological image-based diagnostics into molecular diagnostics with the rise of cell-free DNA for cancer, as well as exosomal profiling for cancer and various other diseases, where you can assess people's likelihood to respond to certain treatments. You can proactively put them on different treatment paths based on their molecular profiles, and also hopefully moving away from invasive tissue biopsies in order to obtain the molecular data into peripheral so-called liquid biopsies, where you're looking at urine, CSF, spinal fluid, and blood plasma and serum and saliva as proxies for or replacements for doing tissue biopsies. Especially in early stage diagnostics, it's helpful not to have to cut pieces out of people or go in with needles. So the advantages of this are, as I mentioned, that liquid biopsies are minimally invasive. They overcome a decent amount of heterogeneity potentially in tissue sampling. Depending on where you sample from a tumor or a tissue, you may find variability spatially within those cells. You can access potentially very difficult to otherwise access tissues via a liquid biopsy. So, prime example is sampling tumor material from the prostate is very difficult and extremely invasive, and leads to its own set of complications that you can potentially get around by looking at markers in the urine, for example. Because it's easy to do, and these, especially using RNA, these assays are cheap. You can, it allows you to do fairly high frequency longitudinal tracking, which is great for looking at drug responses, treatment responses, and prognostics. Equally, you can easily stratify patients into different disease subtypes or treatment groups, and potentially, especially with exosomes and extracellular vesicles, which I am a bad EV person, I will use both of those terms completely interchangeably, which is probably not ideal, but I'm too stuck in the mud now to change. Potentially because these EVs are released in a metabolically active way by living cells, it potentially enables earlier disease detection and diagnosis than you would otherwise get with cell-free DNA or some other molecule type that tends to be released by cells in the process of them dying. That's the hope, at least. So exosomes and EVs specifically, as I mentioned, they're released in a metabolically active way by all cells into almost all biofluids that we've chosen to look at have yielded exosomes and EVs and RNA within them. They're produced by almost all of your cells, and I think an important point is that they can often deliver molecules or expose molecules to the kind of extracellular space that would not normally be secreted by cells. Where usually when you think of things being secreted from cells, you're thinking of proteins, metabolites, and whatnot. And exosomes contain a fairly healthy cargo of RNA and potentially DNA that you would not otherwise be able to see without them. And they, potentially also play various roles in different diseases. They stimulate tumor growth, inhibit immune cells, and various other long-range potential functions where you have a EV released by one cell and taken up into another. So in terms of the open problems, the first category being the sample quality, the RNA quality, and the way that we're isolating and even defining these EVs is a problem. Both of these problems actually, I'm quite glad that more than half the audience is experimental, because both of these problems stem from, the solution is going to be largely as much experimental, at least, as it is computational. Part of the issue with eVRNA is that the quality of it does vary quite dramatically, especially where we're talking about small RNA sequencing, where you're subjecting molecules below 200 nucleotides-ish to high throughput sequencing. You can see that there's a big spread of the types of RNAs that you can detect, as well as the types of contaminants that you will detect in different samples in different biofluids. So, we typically see a large amount of ribosomal RNA, which is very abundant in normal human cells. It's also typically quite abundant in extracellular RNA as well, especially in exosome samples. You can see here from on the bottom left, I don't know if you can see my pointer, but the nice publication from Kendall Jensen's group in 2017. You can see that even though the majority of samples do pretty well, there's still quite a high variability that's biofluid dependent in terms of the RNA-seq signal that you can attribute to the human genome. Obviously in plasma and urine is a significant amount higher than saliva, which tends to have contributions from non-human sources, which is interesting in its own right. But also just in terms of the biotypes of RNA that you're detecting within each of these biofluids can be very different. So, typically in plasma, we see a dominance of this yRNA. Some samples are dominant for microRNA. In urine, this flips completely, where you have actually a majority contribution usually from tRNA and various ambiguous fragments of longer RNAs that have been chopped up. And again, some small contribution from microRNAs. And saliva, very little. Most, a large fraction of this is exogenous. And so, part of the reason that I'm talking about this is because, this became obvious fairly quickly to us when we were looking at the data from the ERCC1, the first consortium project spitting out RNA-seq data. And so we, Joel Rozowski and I, while I was at Yale, we developed a pipeline that would be fairly stringent in how it was handling these data sets in order to try and remove as much of this, try and compensate for this variability as best we could. So we were very aggressive in filtering out likely contaminants from ribosomal RNAs, laboratory contaminants, etc., before mapping to endogenous, and then not shown here later exogenous sources where we're looking for bacterial contributions, as well as contributions from things that you may eat. So one other thing to mention, and another motivation for designing this small RNA-seq pipeline the way that we did, was that, again, and I think Juan Pablo is actually much better positioned to talk about this than I am, but we noticed fairly early on that we had a fairly strong preference in terms of the types of small RNAs that we were interested in quantifying and which ones we believed were going to be in these samples, starting with microRNAs and tRNAs, and then heading down the pecking order into piRNAs and circular RNAs. And so we set up the pipeline such that it would prioritize alignments against microRNA, say, over a piRNA, or prioritize an alignment for a tRNA over a circular RNA, just because the quality and the, I don't know what the word is, but the completeness of the annotation is typically better for microRNAs and tRNAs. And what we found is if you remove certain filters earlier in the pipeline, so if you take away the ribosomal RNA filter up here, if you take away the UNIVEC or quality filters in here, you tend to see now a lot more reads mapping to piRNAs. And so what we found was that actually, about 3/4 of the piRNAs that you would detect if you didn't do these filters are probably actually just human ribosomal RNA fragments that have been mischaracterized, which is not really a surprise because a lot of the experiments that have been done to make the piRNA references are done using small RNA sequencing. So, these reads have just kind of crept through some filters in an earlier publication, made it into a database that then classifies them as piRNAs, and simply, probably, I would argue in most cases, it's not real signal. So we were quite careful to remove as much of this as possible. Joel is probably going to talk about this at length, so I'm not going to go into a huge amount more detail on this. But be a little careful with some of the annotations that you're using and the belief that you have in them. RNA that you get from vesicles is typically quite fragmented and degraded, especially when we're talking about long RNAs. So these are RNA fragments longer than about 200 nucleotides as defined. We see that there's still a lot of ribosomal RNA. You can see here, this is an experiment that we did a while back where we had 2 time points for each of about 14 individuals, and majority of the signal in each of these individuals is human ribosomal RNA. A decent amount of protein coding signal too, but it's variable, and it seems to vary a fair amount by individual, which is also potentially quite interesting. So that there may be some consistent inter-individual effects that are leading to these differences, like global differences in the biotypes that you're detecting in these samples. mRNA, we know, tends to be fragmented in these extracellular vesicles. I think the jury is still a little unclear on whether there's intact transcripts. I think when I was a scientist at Exosome Diagnostics, we did see potentially some evidence for full-length GAP-DH. If that's useful or relevant, I'm not quite sure, but high abundance mRNAs may exist intact within EVs, but I think the norm is for these RNAs to be fragmented, which presents its own challenges for quantification, for looking at reads that span exon junctions. We can see here that the end, if you look at each transcript normalized to bins of 1%. Most of the signal in the preps we were looking at were heavily 5' biased, and that this was biofluid dependent. So urine tended to be more fragmented than plasma and serum, and actually CSF does best on this, surprisingly. But that presents its own challenges in, as I mentioned, if you hope to look at individual transcripts rather than just gene loci in these data, and the expression of genes is fine. But if you're looking at isoforms and transcripts, it gets very difficult because you're relying then on the differences in the annotation between 2 or more transcripts being manifested in the 5' end here, where you would see reads actually mapping to the different isoforms. Sometimes these it's not always a bad thing that these fragments exist, and in fact, there's a growing literature on looking at tRNA fragments that may be functional and released by cells either in a deliberate or kind of housekeeping fashion. And these fragments, depending on where they are cleaved off the tRNA, may have specific functions that the cell does not want them to expose within the cell, or may in fact be trafficked to other cells nearby. Just to mention, so it's also true that most of the exosomal RNA or extracellular RNA samples that we as a consortium have looked at do cluster by biofluid, which is a relief. So you can look across many different samples. You can look across the same samples prepped in the same way by the same groups, and the predominant variability here will be between biofluids, which is good. But the second most prominent source of variability, at least in plasma and serum, is by exosomal isolation or EV isolation method. So you can see a number of different kits and techniques used to isolate the EVs and then to extract the RNA from within them. And the clustering is pretty strong here between each of these kits, which presents its own set of challenges, because it's difficult then for a community to come together with a single database across multiple different platforms that is consistent enough to be useful for as a benchmark for any other platform. You know, it leads itself to questions about batch correction and normalization, which is difficult, or looking, or you know, combination of that and some community agreement over standardization on one method or one type of method, which is, again, it's difficult because each method has, and Louise will talk about this, I'm sure, but each method has specific advantages and disadvantages, and depending on what you're interested in studying, you may want to choose one method over another. So it's difficult to enforce this kind of standardization. It's actually easier when I was at Exosome Diagnostics, and those guys before I got there created the exRNA kit that was licensed out to QIAGEN. You know, it's easy for a company to standardize on a kit. And actually it helps because it allows you to, you know, go back and forth through past experiments and future experiments and have an easier time of it, or at least it made my life easier, comparing data sets historically within the company, because you standardize on a kit and a standard operating procedure. But for a diverse group like the ERCC, it presents some extra challenges. So I think it would be interesting to talk a little bit about ways to correct or normalize for these big differences between kits. That may also come down to the types of vesicles and our extracellular RNAs that they capture, depending on the characteristics of the method. Which leads me on to the tissue of origin, because both are kind of related problems. It's worth pointing out, there was a nice review actually, I think from Zandra's group, Zandra Breakfield's group, just published late last year, talking about, actually mostly talking about EV therapeutics, but a nice figure here shows the types of EV release mechanisms that may lead to some of these differences between kits, and between, you know, the interpretation of the RNA that you're measuring may change depending on what the release is from the cell type that you're interested in of these vesicles. So you can have direct budding in a viral-like sense off the plasma membrane. You can also have vesicles themselves budding directly off the plasma membrane, as well as the classical exosome release from the endosome. The budding off the plasma membrane is an interesting one because, you know, it's the hope, and I'll talk about this if there's time in a few minutes. Just the hope is that, you know, if these vesicles are released off the plasma membrane, there may be some deposition or donation from the parent cell of specific membranous proteins that you might be able to use to isolate via immunoprecipitation some of these vesicles, which may, if you have markers that are enriched or specific to the cells that you want to study, may make your life easier when you're hoping to try and clean up some of the signal of the, especially in plasma, of the total plasma exosome. So the goal, computationally speaking, for deconvolution is to try and figure out the fractional contribution of to whatever biofluid that you're measuring. So you're measuring a total amount of a mixture of something in a biofluid. You're hoping then to look at the fractional contribution from all of the tissues or EV karyotypes that you know about to that biofluid. And so, you know, initial efforts in Alex Milosevic's group, as well as some work at Yale while I was there, and Joel is still doing this as well, to try and deconvolve the exRNA profiles using non-negative matrix factorization, basically just factorization that doesn't allow negative contributions. So you can't have a tissue, although, you know, you can't have tissues that soak up EVs. Everything has to be a one-way donation into the biofluid. And all you're trying to do here basically is say, well, you have a measured set of transcripts in your biofluid, each and you know, n different samples that you have in your that you've measured the transcript abundance in, say, blood plasma. You know what the expected expression profile is in a number of cargo profiles here, different carrier profiles, or in different tissues or cell types. So you know what these transcripts look like in each of these tissues, and then you're just trying to figure out what the matrix looks like that you need to multiply this to get this. So you're just saying, how much of each of these tissues do I need to mix together in order to see the profile that I've measured in the biofluid. And to an extent, it works in a crude way. It's difficult, as you can imagine. One of the primary things that is necessary to have high confidence in is this matrix here. This matrix here, where you need to understand exactly what the transcript abundance is in the tissue or the cell type or the carrier profile that you're hoping to measure. Because if you aren't confident about this, then there's no way you're going to get a very good estimate of the proportions. Again, what Alex's group noticed in the Cell paper that was published for the Atlas was that there is a big difference in the kind of deconvolved carrier types between different kits. And as I mentioned, this may just simply be that different kits have different characteristics for different reasons and will be potentially catching different types of extracellular RNA. And as I mentioned, this may not always be a bad thing, but it's worth thinking about before diving into the full Atlas data set, for example, because certain samples may be more difficult to compare to each other than others, depending on the types of kits that were used to generate them. And I'm not convinced that it's going to be possible, frankly, to do deconvolution with plasma and serum in a way that is useful. Especially in a diagnostic sense. It's extremely difficult to think of the FDA giving any kind of approval to a diagnostic test that involves a computational deconvolution. So the task here is to understand enough about the biology to help refine the biomarkers that we're then taking forward into clinical diagnostics validation set. I don't think it's, because the deconvolution requires you to always measure all the transcripts that went into this original signature matrix. You always have to measure the same N, roughly speaking. It doesn't lend itself very well to a diagnostic. But again, happy to argue that with people, and especially in the company that I was at, we chose to go, the PCR route was the best because it's understood by the regulatory folks better than NGS tests, and also it's cheaper. So, as I mentioned, when you're doing the deconvolution, you're hoping to overcome a mixture in a biofluid, and you can see where this comes from, right? So you have, for example, some disease that you're hoping to measure through measuring plasma EVs. You're hoping to resolve any differences in RNAs that are, the origin is the disease tissue or cell type, against a massive background of EVs emitted from healthy tissues. And plasma is a difficult biofluid too. I mean, if you think about urine and spinal fluid and saliva, they tend to be kind of waste biofluids, whereas plasma and serum are circulating. So in some senses, it's actually easier to think about measuring a release of exosomes into a biofluid that's a waste product. But in the plasma, there's a lot of contributions from a lot of different tissues, only a fraction of which derive probably from the disease. Especially in an early stage disease, this just gets harder and harder and harder as your contributions are smaller and smaller. And so part of the focus of ERCC2 and of the company when I was there was and various other groups that you'll hear from over the course of the next couple of afternoons or evenings, depending on where you are, is to use biochemical enrichment or depletion, where you can look at surface proteins that may be donated from the parent cell, that you can go in with antibodies and just try and clean up some of the background, especially in the enrichment sense. If you can understand where these RNAs are coming from, it may give you an easier route to, the deconvolution may get easier, and also just finding disease-specific signals amongst this background might get easier. Oh, Alex is correcting me. All right, we can discuss that in a bit. So, I think I'm running out of time, so I'll whiz through this quickly. This is the last slide. So if you think about this visually, as I tried to do, if you think about the complement of RNAs that in a certain disease, say it's bladder cancer or breast cancer or leukemia or any, anything really, you have a cohort of RNAs that vary by disease. People may have found these and published them in tissue profiles or cell culture. And then you can think about the overlap of those RNAs with RNAs that you can measure in plasma EVs that are predominantly contributed from the target cell type, so be it the tumor cell type or whatever. Given this high background from other tissues, it's actually quite rare to find RNAs that satisfy both these conditions. It's possible, and actually cancer is one of the easier diseases to study for this, I think, but it's possible to find them, but they're in the minority for sure. And so the hope, I think, is that using an enrichment, you don't do anything to this side of the Venn diagram, obviously, because the RNAs that are varying by disease don't change, but that the RNAs that you can find in your biofluid that can be, reliably thought of as being derived from your tissue of interest or your cell type of interest gets higher because you've removed some of the non-specific background. So this presents a bunch of other challenges, some of which are computational, that are interesting. Is then what is the optimal intersection between a cohort of proteins that you can go after with antibodies and RNAs within those EVs? You have a nice little set of logic gates that you can build experiments to satisfy, and what combinations give you the best purification of your disease-specific signal. And this also has implications for, it makes it very important to look for potential normalizers, as I'm sure we'll talk about later in the session, that may adjust for and compensate for differential efficiency in this enrichment process, because the yields can be very different. I think I will leave it there. Roger's encouraging me to stop, so I will stop now, and we can address questions later.